This is episode 81 of the Illinois Nutrient Loss Reduction Podcast. I'm University of Illinois Extension's Todd Gleason. Today we'll explore the role of the soil microbiome in reducing nutrient loss with a couple of folks from Waypoint Analytics. We'll learn about it and Elizabeth French and Mick Goedeken as we explore the biome and how it interacts with the ecology of the soil. Let's start with Elizabeth French. She goes by Lizzie. Hi, Lizzie. Thank you for being with us. Tell me about yourself, please.
Yep, I'm Dr. Lizzie French. I currently live in Laramie, Wyoming, but I was spent a good chunk of my adult life in the Midwest. I got a biology degree at Indiana Wesleyan and then went to grad school at Purdue in the botany plant pathology department. I started working on projects with tomato pathogens, but then I got really interested in all the other bugs that were out there in the soil and really ended up focusing more on just the soil microbiome as a whole, how it interacts with the plants and how that ends up impacting plant health and soil health. And I liked it so much, I stuck around a couple more years and did a postdoc in a different lab at Purdue, and then ended up moving into the industry, worked with Nutrien for a few years on soil biology and how we can use it to make agronomic decisions, and then moved over to Waypoint about a year and a half ago now, and have been loving it, really enjoying the work that I've been able to do with them, looking at soil microbes and how we can help make agronomic decisions, understanding, having that fuller understanding of what's going on in the soil.
Mick Goedeken is here too. He's a regional agronomist with Waypoint Analytical. Hi, Mick. Thank you for being with us. Thanks for joining the Big Ten. I see you are at Nebraska or a Nebraska fan. So we have the U of I along with Purdue. We're only 90 miles apart, so we're kind of close. Right. Yep. You're way out there in what we call the Western Corn Belt. But tell me a little bit about yourself, please.
I'm Mick Goedeken. I have a master's degree in soil fertility. I started out my professional career in the soil testing world, and then I decided to move around a little bit, and I've experienced retail agriculture. I've experienced wholesale agriculture, the seed industry, and then I worked in research. And then I most recently, before joining Waypoint 2 years ago, I actually worked for Orthman Manufacturing and was their the agronomist for Orthman Manufacturing, covering the entire globe. So now, having joined Waypoint, I have a territory that is from Idaho to Ohio, so it's a much smaller than the entire globe. And most of my counterparts call me the Corn Belt agronomist. I cover the 3 labs in Grand Island, Nebraska, Champaign, Illinois, and Atlantic, Iowa. So I am the agronomic support system for those 3 labs. And then I also support the sales efforts and try to learn from Lizzie as much as I can.
Lizzie, on that point, tell me about Waypoint Analytical. What is this organization?
Yeah, Waypoint Analytical, we are one of the largest ag soil testing networks in North America. We've got 7 different ag labs across the U.S. And we run probably 2 million plus soil samples in a year. But we also offer, tissue testing, water testing, manure, fertilizer, pathology testing, kind of disease diagnostics, nematodes, and now soil biology testing as well, for looking at more of the good guys in the soil, not just the pathogenic organisms.
We're here today to talk about the soil microbiome. Can you tell me what that is and why it's important?
So the soil microbiome is everything, all the living organisms you can't see in your soil, right? So you've got bacteria, you have fungi, you have viruses, protists, nematodes. There's thousands of different species of organisms. And, if you pick up just a spoonful of soil, there's going to be billions and billions of these tiny microscopic organisms living in your soil, making up that soil ecosystem, and doing a lot of really important jobs in the soil that we don't always think about just because we can't see these organisms. They're really important for nutrient cycling, mineralization of nutrients, breaking down residues, building up soil structure, all really important things that we need to happen in our soils from both from a production standpoint and a conservation standpoint, right?
Mick, I'll bet when you talk to producers, maybe even to some of the folks in your lab, they ask you, hey, why is it that this soil microbiome thing you're talking about is important to us? Can you put it in terms that I can understand as a farmer who's just wanting to know what NP and K might look like on my soils?
So for years as producers or agronomists, we've focused on the chemistry aspect of the soil and getting the soil chemistry correct. And then we started looking at the physical properties and saying, okay, now this biome portion is bringing everything. So we have the biological, the physical, and the chemical all included in it, so that we can see exactly what that soil can do for us and make us more efficient. Some of the things that the biome tests allow us to see is how many microbes are out there that can help facilitate keeping nitrogen in an available form. Nitrogen is a very complex cycle and is biologically driven. If we have the wrong microbes in there, we're going to cause a lot of denitrification. Well, we don't want denitrification to happen. So if we know more about our soil, we can know more how to treat that soil and how to apply our nitrogen more efficiently.
Lizzie, this is about the biology, the mechanics of the soil, really the ecology of the soil itself and how it works together. What do you think creates a healthy soil across the Corn Belt?
Really, like Mick was saying, it's those three components together, right? You need to have the soil structure that allows you to have good air infiltration, good water infiltration. That interacts, the soil biology plays a role in that, where you've got, microbes producing these sticky glues that help kind of build that soil structure. You need the chemistry, you have to be able to grow a crop. That's what soil health is in production agriculture, right? You got to be able to grow a crop, and you want your grandkids to be able to grow a crop too, right? So you need to have the chemistry there to be able to grow that crop. And then, yeah, like I said, the biology piece interacts with all of that. So you need to have a healthy, thriving community of microbes that are able to cycle those nutrients into available forms that are helping to build that soil structure. And all of those things affect each other. So they you can't really pull them apart to understand them. You have to look at them as a whole to be able to really get an idea of the whole system. And so really, when you're talking about the Midwest, corn and soy, having, sufficient levels of the nutrients that you need to build those crops, having solid soil structure, and then having high levels of soil biology that are able to then, make those nutrients available, keep that soil structure where it needs to be.
Well, get to making the nutrients available or keeping them in place, probably both, actually. But how is it that you measure a microbiome? What happens? And are there good things and bad things about it?
Yes. So there are lots of ways to measure. The thing most people are probably familiar with is something like the Haney test, where you're getting a respiration metric. That is really kind of a 40,000-foot view of how much overall microbial activity do I have? Are they living and breathing? And that's, that is a great high-level metric. It doesn't tell you specifically what those microbes are doing and what they're capable of doing in your soil. What we've done with our test is we're looking at the DNA of the microbes in the soil. So you can think about, when you do a nutrient extraction, you have a specific process where you're separating out those nutrients, and then you're able to kind of analyze them on an instrument. we're doing the same thing, but we are finding the microbes in the soil, taking their DNA, and then we're able to specifically look at that DNA and say, okay, how many of you are nitrogen fixers? How many of these microbes in the soil can solubilize phosphorus? How many of you are mycorrhizal fungi that are going to help bring, essentially extend the root system and help that crop acquire more nutrients? So that's what we're really able to answer using that DNA analysis, is kind of who is it that's there and what are those microbes actually capable of doing in the soil?
On the microbiomes, are they capable of keeping nutrients, particularly nitrogen, phosphorus, in place? And if that is true, do they lock them in place so that they're not usable by the plant as well?
That's a great question. So, microbes need nitrogen and phosphorus too for their survival, right? So one of the ways that they help to keep it in the soil, well, there's two ways. So we'll talk about the first one. The first one, they keep it in their biomass. So as part of their body, they're keeping nitrogen and phosphorus. When those microbes are growing, yes, they do temporarily lock up those nutrients so that they're not available. But microbes have much faster life cycles than we do or that plants do, right? So that, within a short amount of time, days, weeks amount of time, that those nutrients that have been kind of vacuumed up into the microbial biomass become available again as they go through their life cycle. You have other organisms in the soil like protists that are kind of tiny little lions keeping the population of the antelope in check on the soil savanna, right? So they're able to go out and eat those bacteria, then make that nitrogen available again for the crop. So that's the first mechanism that they can use to keep nutrients in the soil. The second one is, which most people are probably more familiar with, is through building up organic matter. So that microbial activity, a lot of that microbial biomass, those dead microbes, become part of the organic matter. Those microbes are important and they break down residues. They keep that carbon in the soil. A lot of those nutrients are kind of locked up into that carbon. And so the soil organic matter, we really want it to be dynamic, active. You're building up new organic matter as you're mineralizing some to be able to make new nutrients available. So it's this cycle that microbial activity keeps going, where it's able to then retain those nutrients in the organic matter, make them available again for the crop.
Mick, you're an agronomist. You're also a farmer. When you talk to producers after they've maybe taken a look at what their soil biome is like with Waypoint Analytics, how do you tell them about deploying that biome and what practices will help them to do so?
The best way I can describe it is we take the information that is in that biome test and we can manage our individual fields more successfully, is the way that I would look at it myself. If I know a field has a lot of phosphorus mineralizing bacteria in it that's going to make phosphorus more available, then I can be assured that I can manage that phosphorus more efficiently in that field. If I don't have that, then how do I get to there? And I'm a true believer that a lot of the bacteria that we need in our soil is there. We just haven't fed it the right way to get it to where we need to get it to. And so then maybe it's a practice where we change what we're doing so that we can build the bacteria there.
So Lizzie, you're maintaining that it's important to understand how the soil biology interacts with, I guess, the fertilizer management. I think that's right.
Yep.
So can you improve the biology to manage that fertilizer?
Mm-hmm.
And how do you do it?
Yes.
What are the biggest opportunities, you think?
Mm-hmm. Yeah. So from a big picture perspective, right, just any practices you can implement, this part is I would say it maybe gets rebranded every 10 years or so, but it's still it's the same the same principles. You can boil it down to, your microbes need food and they need housing. And if you think of that, from a soil health perspective, that's protecting your soil structure, so minimizing tillage as much as possible. That's going to prevent you from breaking up all of those nice fungal networks that spread out throughout the soil and help your crop get to nutrients. And it's going to help, protect the soil structure, protect the housing for those bacteria as well. And then you need to feed them, right? And whether that is through organic inputs like manures, compost, all of those things provide carbon to the soil for those microbes to live. Cover crops, just keeping a living root in the soil for a longer period of the year, where you're able to then feed that soil biology longer. All of those practices are going to build up those populations of microbes overall, and help you do more with the fertilizer that you're adding, because you're able to then, soak up that fertility into that microbial biomass that you're feeding. You're able to, you've got populations of solubilizing organisms and mineralizing organisms that are going to help, if those, say, phosphorus is getting tied up in your soil, you have that microbial activity that can help kind of bring it back into solution. Versus, if you have lower levels of biological activity, that is, you'll need to apply more fertility to get the same result in your crop.
How important, Lizzie, are conservation practices in this soil biome, in this whole ecology of the soil and its health? You mentioned cover crops. I'd also like to know about kind of reduced tillage kinds of things, maybe waterways, the kinds of things that move water through and out of the soil. It could be even tile. You tell me what's important.
Yeah, so all of the above, I would say. The tillage, again, you don't see huge increases in soil biology as a result of reducing tillage, but it maintains what you have. You don't end up losing. And then adding in other practices like cover crops or application of organic inputs is going to build that soil biology. Waterways is an interesting one. Like I mentioned, I live in Wyoming, so we have the opposite problem of not enough water to keep our microbes and our crops happy. But yes, in Illinois, in the Midwest, where you have too much water sometimes and you need that tile drainage, that's actually a really important component of managing, a part of the soil microbiome, a part of that nitrogen cycle that leads to nitrogen losses, right? So nitrification, denitrification, those are both processes that happen entirely because of microbial activity. So you have the microbes that are taking that ammonia, converting it to nitrate. If you have a lot of rainfall, that nitrate can end up leaving through your waterway. You could end up getting denitrified by another group of organisms that are going to take that and convert it back into air, essentially. And so you've completely lost your nitrogen out of your soil that way. And those are all microbes that, you can manage through tile drainage, through practice, like applying a nitrification inhibitor on your nitrogen, splitting your nitrogen application so that there's not so much food for the microbes to then go chew up and get rid of at one time. There's lots of, things that you can think about from a nitrogen management perspective, especially when you can see one of these reports and see, I have a really high potential to lose nitrogen, given the right environmental conditions. So I need to think about how I'm going to be protecting that nitrogen investment that I'm making on that field.
I do want to narrow this down a bit. So we could talk about the Corn Belt from western Illinois up through parts of Iowa, even into Minnesota. There's a large portion of that part of the Corn Belt which has similar soils, relatively speaking. How should maybe they manage their soil biome, do you think? And Lizzie, why don't you start for me?
I would say this is always a tougher question, right? It's much easier to answer this question for like, say, the South Plains of Texas, because, then really the soil there needs all the help it can get. Whereas, in the area you just mentioned, Illinois to Minnesota tend to have, yeah, those really nice, like high organic matter, really good soils. So in my mind, and we typically see when we look at those soils relatively high levels of overall soil biology, even in, conventionally managed fields, just because you've got a lot of food there for your microbes. If you've got, 4 or 5% organic matter, you're going to have a lot of biological activity. And so I would, in those cases, definitely the conservation piece is important, because if you're not keeping that soil where it is, you're not going to have that microbial activity and good, healthy soil forever if you're not doing what you can to conserve it.
How about I jump in there and add,
Yes, please do.
because that region that you're discussing experiences a lot of compaction.
Yes,
And
yes.
when it comes to microbes, compaction is an enemy.
Yes,
And
absolutely.
we don't give compaction enough credit for what it does to the plants. And if you think about, I wish I had time to go through my presentation on corn roots and the push power of corn roots, because corn roots don't have a lot of push power when that corn plant is little. And most of our compaction is at a 6 to 8 inch level across anywhere in the world, but especially in that area, because we end up going into the field when it's not ideal conditions, because we farm on a calendar system, unfortunately. And so we'll make passes where we shouldn't, and we create compaction. And when we create that compaction, it hurts the soil biome. It really does. And so managing that compaction is something that needs to be done. And we talk about reducing tillage, and I did work in the strip till world, and I'm a fan of strip till as long as it's a straight shank and that we're getting below that compaction layer and actually breaking that compaction layer up. Those are things that are very important.
When we talk about the soil biology and the interaction and fertilizer management, as we've been doing some, how can you or a producer improve the biology to reduce fertilizer needs?
Yeah, again, so improving your soil biology in terms of specifically for nutrient cycling, it's still the similar practices. Those microbes are there. They might just not be at the level that you need them to be able to provide those benefits. And so implementing those conservation and soil health practices is going to help build up those levels. If you have, a soil biology test in hand and it's telling you that you already have high, good levels of soil biology, then that's also a point for you to trigger to think about, if I have decent levels of organic matter, I have high levels of soil biology, maybe I want to consider dialing back that nitrogen fertility a little bit, because I can pretty confidently say that you will not see it. You're likely not going to see a yield loss if you have that organic matter to mineralize, you have that soil biology, you're probably gonna, and I hate to make a hard and fast recommendation for how much to dial back, just because that does depend, heavily on how much rain you're getting, things like that. But that is definitely something to take into consideration when you're thinking about fertility, especially in the environment we're in with how the cost of fertility at the moment. This type of information can help you feel more confident about your decision to dial back on fertility if you have that capability to provide provide those nutrients from your soil and from your microbial activity.
What is it that you would like to leave with farmers? What kinds of things should they understand about the soil biome, the tests that you provide at Waypoint, how they might deploy it? And Lizzie, I guess I'll start with you.
I would start with that, whether you've realized it or not, you've been farming your microbes this whole time. And they've always been a part of the agricultural system, whether we recognize what they were doing or we didn't. And they're a huge component, and it's a big blind spot if you don't know where you're at as a system in terms of your levels of soil biology. So I would encourage folks to just to take a look, try, just one or two samples across a field that's got maybe some variability in yields, where you can maybe try to understand, hey, is this low yielding area on my field that maybe is normally on a high yielding field? Is that a biological limitation, or is there something else going on there that you might be able to identify with this type of testing, and just be able to think through better how to use the fertilizer you're applying most efficiently when you have all of the information available about your field.
And finally, because Mick, this is in your direct wheelhouse, are there things farmers should consider before getting a soil biome test?
Yes. Consider learning from what you've done, and consider learning, and don't be afraid to change, because when you do this biome test, you're going to learn some things. Not every field's the same. A blanket treatment of your entire farm doesn't work. If we start managing field by field, I think we can improve efficiency a lot.
Anything I didn't ask that I should have, or any other final comments from either of you?
Oh, the last thing I remembered, we talked a lot about conservation practices and soil health. The other big thing that we did not talk about from the soil biology perspective was pH. If your soil is very acidic, your soil health practices are not, like, they might help a little bit, but you're still gonna be limited by that pH in terms of your biological activity. That is one of the biggest drivers of soil biology in the soil. And so if your soil has become acidified over time, that's your first step. You need to manage that first, and then you can work on the other components to build up soil health.
So Todd, remember when we said everything was related, right?
Yes.
Early on. So pH is the driver for biologicals. It's also the driver for the chemistry.
Yeah.
And the efficiency of that chemistry. So once again, we just tied everything all together again.
Mick Goedeken and Lizzie French are both with Waypoint Analytics and joined us here on the Illinois Nutrient Loss Reduction Podcast. This has been episode 81, The Role of the Soil Microbiome in Reducing Nutrient Loss. Our program is produced in conjunction with University of Illinois Extension's Rachel Curry, Nicole Haverback, and Luke Zwilling. I'm Todd Gleason.